everybody hear me?
Yeah, we sure can.
Okay,
It's
great.
my pleasure to introduce Dr. Irene Salinas. She's a professor within the biology department at the University of New Mexico. She did her PhD at the University of Murcia in Spain on fish gut immunity and probiotics. Her research interests are fish mucosal immunology, host microbiota interactions, fish vaccinology, and neuroimmunology. And she's also a strong advocate and active ally for equity and inclusion in STEM. Well, thanks a lot for presenting today, and we're looking forward to your talk.
Thank you so much for hosting me and inviting me and for putting together this amazing series over the last few years. I think it's doing huge benefits, bringing huge benefits to our community and for everybody to interact on Zoom. So, okay, I just want to start thanking a lot of the people through the years who have been in my lab, and I think it's important to thank them first. All of these people have taught me a lot, and I've learned a lot from their diverse minds, and I just want to thank everybody already from the beginning. And a lot of the work that I will present today on nasal immunity has been done by Ben Garcia in the lab. We also have Ali, who has just started recently, and he's taking over also this project. So why I love mucosal immunity is because I always see that this is the hardest part to understand in terms of the immune system, which is already very complicated, but mucosal barriers have to interface all the time with the environment. And as we know, this is incredibly important in the context of fish health. And water quality is always one thing that we worry a lot about. And of course, that's impacting all the time, not only the physiology of mucosal barriers, who have incredibly complex physiological functions, as well as the immune system that is associated with these barriers. And today, I want to, I guess, explain some fundamental concepts about mucosal immunity and all of the major breakthroughs that are happening in the field right now. And how do I really believe that we still know so little, and that basic immunology is incredibly useful, because those principles is what should guide us when we decide how and when to vaccinate fish for improving fish health. So this is just an introduction to how the mucosal immune system is organized in mammals. And we know that we have two major sites that are defined by the Society for Mucosal Immunology, that they tell us to use this nomenclature. And these are the very well described inductive versus the effector sites in mammals. So the inductive sites are constantly being exposed to antigens in the outside world. And these antigens are, here you can see one on the left-hand side, are constantly stimulating the formation of or stimulation of naive B and T cells in very well organized structures, and there is the generation of memory at those sites. Those cells that have been now encounter antigen in these inductive sites, where the immune response is truly or really initiated, they will then enter the lymphatic system and then reach what we call the effector sites, where they will, of course, deploy their effector functions. And you can think, for instance, about IgA B cells. They will then get to this lamina propria in the gut and then produce and secrete IgA, and then that is their effector function. So for many years, since I started my PhD, we have really not been able to understand well how this organization happens, or whether it even happens in fish. And of course, that poses a lot of big questions about how mucosal vaccines work in fish, because truly in mammals, mucosal vaccination is designed or targeted to stimulate these inductive sites. So in mammals, we do have very well described areas within the germinal center reaction. And one thing I would say is that these germinal centers here at mucosal sites is what mammalian immunologists call chronic pair patches, because they are all the time receiving microbial antigens, as you can imagine, from your gut or your nose, etc. And those are very different from those found in the spleen, for instance. So all of this is very well described in mammals, and what we know in the germinal centers ultimately is that there is going to be a selection of B cells. So we are going to eliminate the cells that are not good, or that they are not the best for the antigen that they just encounter. They will also produce long-lived plasma cells, and there will be, like I said before, the generation of B cells. And that is an incredibly complex series of steps that goes from the centroblasts, which are the very first B cells, and then they are going to undergo rounds of many bursts of proliferation and somatic happy mutation to get the very best, most strongest affinity B cells possible. So I just want to start putting together a bit of history of what we know about mucosal immunity in fish and in the context of aquaculture. Is that we know since 1940, so that's also going to be 80 years ago, that fish mucosal vaccines work, and the very first one published out there is in 1942. It was an oral vaccine against Tyromonas salmonicida. A very, very seminal work at the end of 1960s in this paper that I absolutely love by Fletcher and Grant. This paper really showed that there are immunoglobulins that are detected in the mucosal secretions of plaice. And later on in 81, another really seminal paper by Love and Clem, where they show that these immunoglobulins that can be detected in the mucus are actually the result not from transudation coming from the serum, but actually from local production. And they experimentally demonstrated that in a very elegant way. So this told us that there is a mucosal immune system in fish, and we know that we can at least try to manipulate it to our advantage to improve fish health in the context of aquaculture diseases. I started my PhD in 2003, so a long, long time ago. And one of the things that I want to do throughout this talk is really highlight all the beautiful things that happen during your scientific journey, and not just the discoveries in the lab, but also the people that you meet on the way. I want to say that always the best parts are meeting friends in conferences, and these are one of my really great friends, Sonal and Ingrid. And I always say, find the people in conferences that make you laugh the most. And some people in the background maybe recognize themselves. There's Niels there. But just have a lot of fun in conference and just hang out with those people who bring you all of those laughters, and keep those friendships afterwards. So they're still my friends now. So one of the things that it was very apparent during my PhD reading a lot of papers about mucosal immunity is that we always start the review saying, okay, fish do not have any of these organized lymphoid structures at mucosal tissues. And I just introduced you to that concept because I show you that there are inductive sites and effector sites in mammals, and that those inductive sites are incredibly well organized. They have those B-cell follicles and the germinal center reactions that truly allow the microenvironment for the B-cells to mature and respond to the antigen. Interestingly, right after my PhD in 2008, amazing group in Norway who are incredibly good at anatomy. They discovered interbranchial lymphoid tissue in Atlantic salmon. And this was very exciting to all of us mucosal immunologists, because for the first time we could see that the gills in fish were not just scattered lymphoid cells all around, but they seemed to have at the base of the lamella, they had these massive clusters of immune cells, lymphocytes, that they showed that were mostly T cells stained here with an antibody in brown for CD3. And then you can see all of the scattered cells here on the side. So this is the interbranchial lymphoid tissue. And we started then to ponder if we had been wrong for a while, and whether then, therefore, this dogma should be challenged. I then started my postdoc in 2008 in Dr. Sunir's lab, and he was for a long time looking for the function of an immunoglobulin that had been discovered in 2005, IgT, and probably I don't need to tell this audience the importance of IgT, but this was, of course, a very exciting time in my career where we just basically lived in the lab and slept in the lab until we truly found what this molecule was doing. So I really cherish these years as being really, I guess, special in terms of the depth of my academic training. And I was, of course, always fascinated by microbiota. And so this project really brought all of my interests together, where I had done my PhD in gut immunity, and I was able to bring that expertise to Oriol's lab, and then really started to work on the interactions between immunoglobulins and the microbiome. So how are these immunoglobulins secreted into the mucus? So this is a really beautiful, I guess, biological process where these B cells or antibody secreting cells at mucosal sites are never directly in contact with the lumen, where the immunoglobulins need to reach to protect the host. So there is always the help or the need for a transporter molecule. In this case, we call it the polymeric Ig receptor, PIGR. Some people call it PIGR, and this receptor is expressed down here. You can see in red in the basal pole of these epithelial cells, what is going to bind the antibody secreted by the antibody secreting cells here in the lamina propria, and it's going to via transcytosis across the epithelial cell release a piece of the polymeric Ig receptor as well as the immunoglobulin into the lumen. And this is a schematic from IgA, dimeric IgA in mammals, but we know that this is very similar in fish, as we demonstrated also during my postdoc in the Sunir lab. So what is the functional significance of this microbiota being coded by immunoglobulins and mucosal barriers? And this is a very big question in the field because it seems to have a lot of paradoxical roles. We know that IgA binding to bacteria in the lumen prevents bacterial motility. It also causes clumping via in-chain growth reactions, especially in fast-growing bacteria. And that is then going to allow the elimination of those clumps via just basically the gastrointestinal movement of the gut and then elimination in the feces. We also think our people have postulated that there is a decreased chance of horizontal gene transfer among bacteria, and therefore perhaps coating by mucosal immunoglobulin should decrease bacterial evolution within the gut. But we also, apart from this kind of idea of elimination of the bad bacteria, there's this paradoxical role of SIGA. Truly has been shown in multiple studies that it actually favors or enhances, supports colonization of mucosal barriers. And that can be done either directly through direct coating as well as via several indirect methods. Of interest, especially because in fish, this is also very, very true. Most secretory immunoglobulins are very low affinity and very broadly specific. And several incredible papers in microbial ecology and modeling that have really showed that they should in fact favor slow-growing bacteria, which again is interesting because in fish, especially in cold water species, many bacteria are slow-growing. Of course, that's probably not true when you're thinking of really nasty pathogens in catfish. Okay, so I just want to, of course, show that in IgT, what we discovered in 2010 was that this is a very important immunoglobulin against pathogens. So we showed that it was required for the mucosal immune response against a mucosal parasite, in this case, Ceratomixa sasta in rainbow trout gut. But we also then showed in many, many papers that this IgT is coating microbiota in the gut, the skin, the nose, and more recently, many other papers have shown also in the oral cavity, in the swim bladder, etc. One of the things that is very apparent from all of these microbiota coating studies is that not only IgT, but also IgM is playing a role in coating microbiota. And this is something that we have now under review, is available as a preprint, where we have also shown how incredibly important IgM coating is for microbiota. And also there is a very large proportion of microbiota that are in fact coated not only by IgT single-coated or IgM single-coated, but there is this synergism where both IgT and IgM can be found on the same bacterial cell. And this is a very exciting phenomenon because it also happens in humans, and we still do not know what the functional significance of this double coating means for the host and for the bacteria. A very big question that emerges from all of these studies is how do the IgT-less fish do it, right? How do they maintain mucosal homeostasis and mucosal health when your genome does not encode for an IgT or an IgT-like molecule? And here you guys have a couple of examples, Medaka fish as well as catfish, which we all know that their genomes do not have IgT. So I'm really hoping that somebody tackles this question because it's fascinating. So how does it look like when we just collect mucus, right? Many of us go and sample fish. There are healthy fish and sick fish. And where do we find immunoglobulins in all of the different surfaces and what are their proportions? So what we know is that in serum, the concentration in micrograms per ml of IgM is very, very high, much, much higher than IgD and much, much higher than IgT. But even if this is also true at mucosal secretions, including the gut, the skin, and the gills, and the mouth, and the pharyngeal mucus, their proportions of IgM to IgT are very, very different. So if we do a ratio between how much IgT and how much IgM we have in each of these secretions, what we see is that in all mucosal secretions, this ratio is much, much higher. It can range between 20 in pharyngeal mucus and nasal mucus, all the way to 63 in the gut, compared to a much, of course, inverse kind of relationship when it comes to serum. So all of this really supports the idea that IgT is truly a specialized immunoglobulin that plays a really important role at mucosal sites. So I finished my postdoc, and then I went to New Zealand for the second postdoc, and I was working there on skin immunity and skin parasites in New Zealand grouper, and I had a really great time getting to know another culture and having a van and exploring both North Island and the South Island in New Zealand. And then I was really, really lucky to get an interview here at the University of New Mexico at the end of 2011. I opened my lab, and I have been here ever since. And this is a great photo because my friend from Philadelphia came to visit me, and this is literally this is where I am sitting right now. This is the same office, and this is when my first day at school, I guess. So all these years, like I said, we were just being told that fish do not have an organized mucosa-associated lymphoid tissue. That where are those inductive sites? Do we really even have inductive versus vector sites, etc.? So for all of those years, we wrote many reviews and many papers showing that there's skin, gills, and gut associated lymphoid tissues, and these are just basically loads and loads of diffuse networks of leukocytes that are scattered around these mucosal surfaces. And so in my head, that's what it was. And we kept saying, okay, this is what it is. And we described in 2014 the nasopharynx associated lymphoid tissue in my lab that we use just the mammalian nomenclature NALT. And we just define it as, again, there is no organized NALT. This is just diffuse mucosa associated lymphoid tissues. We just see immune cells scattered everywhere. And these are just some examples from very different fish species. We mostly work in trout, but we looked at goldfish, we looked at catfish, we looked at eels, and every single nose we looked at, olfactory epithelium was really, really teeming with immune cells. And we also showed that basically the IgT to IgM ratio was looking very, very similar to other mucosal sites. So we described this organized nasopharynx associated lymphoid tissue, which is all diffuse, and we just looked at this piece here, which is the olfactory rosette, which is basically the olfactory organ, basically just detecting chemical substances in the environment and really responsible for the sense of olfaction in teleost fish. And we were really surprised because for being such a small organ, and I had worked with gills, skin, and gut before, it was incredibly easy to get a lot of immune cells from there because, like I showed you before, there's just a lot of them. So what our findings did, and this is just basically in support of basic science translating into applications, that we really show how effective nasal vaccination is, and we are still trying to figure out why it's so effective. But in reality, over and over and over again, we show that if we put at least live-attenuated viral vaccines as well as killed bacteria such as enteric red mouth vaccines, we can really confer 100% protection very early on, and also that this protection is very good, at least for a month. We have done longer vaccination studies. We've shown protection up to 6 months. And we were surprised because in the case of IHMB, actually, the live-attenuated nasal vaccines were superior to the injected vaccines up to 6 months. Although I'm not showing you this data here, you guys can find that paper if you want. We do get, of course, a lot of, concerns about, how are you going to be nasally vaccinating individual fish, and do immersion vaccines should already reach the nose? And I'm happy to answer those questions at the end because, of course, it's a long conversation, and we know quite a lot about immersion vaccines stimulating the nose, and also how to apply our findings in much more practical ways for the industry. Okay, so one thing I also continue to do in my lab was really digging deep into the microbiomes at mucosal sites. So in 2015, we map all of the microbial communities in rainbow trout in different sites, and we show how they look like in different parts of the gut, the skin, the olfactory organ, the gills, and how they can also be very effective at controlling fungal pathogens. And we just did some essays where certain isolates from our collection were able to inhibit fungal growth. And so that was really exciting. Okay, so meanwhile, I had an undergraduate student, Cecilia Kelly. She came to my lab. She was in biochemistry, and she said, I want to join your lab. And my question is, I want to know what PIGR does once it's secreted into the mucus, which I told you before is called secretory component. So this is that piece of the PIGR that gets transported across the epithelial cells to release the immunoglobulins to the mucus. And he said, I want to do this project in your lab. And I said, great, because I have had that question for a long time. And I love the fact that you were able to come up with that question yourself. And Cecilia was just truly amazing in the lab. She had amazing hands, amazing brain. And I have such good memories of working with her on this project. So we showed actually that free secretory component in the mucus, so no longer bound to the immunoglobulins, because once it's released into the mucus, that interaction is a non-covalent interaction in fish. There's no disulfide bond. And so we were able to show that actually the majority of the PIGR molecules in the mucus are found as a free molecule, not bound to Igs anymore. And we also showed against a panel of bacteria, pathogenic and non-pathogenic, that this free secretory component is really good at coating both microbiota as well as pathogens. And I'm still really fascinated about this. I would love to know more in the future, particularly in cases where IgT or IgM secretion is impaired, whether secretory component can basically take over those roles. And also in the case of the species, like I told you, the species that I told you before that do not have IgT are in those species, perhaps this PIGR coating playing a much more permanent, predominant role. And so we put together a model for microbiota coating where, as you guys can see, we can have microbiota that are coated just with immunoglobulins, microbiota that could be coated by both immunoglobulins and the secretory component. And as well, we showed that there were microbiota that did not have any immunoglobulins that were just coated by the secretory component. And again, the functional consequences of all of these modalities of microbiota coating remain to be elucidated in terms of transcriptomes, metabolomes, etc. What are the consequences to the bacterial cells? We don't know. So we always kept searching where are those sites that we should be able to hit with mucosal vaccines, and really where are those inductive sites, and or whether they exist or not. And of course, it has been a long-standing question in the field, and we have hopefully done some contributions in my lab towards that question. Here, for those who always ask me, do fish have noses? Yes, they have noses, and this is the proof of it. I love fish noses. They look like flowers, and they are great because they are so apically located and externally located that we can vaccinate those really easily and put the vaccines right there in that little hole that is pointed with the arrows. But at the time, we had a student in 2018 coming from Spain and visiting the lab, and he was a veterinarian, and he was incredibly good at anatomy. And he always told me he felt a little bit bothered about the fact that we were putting tweezers in there and taking the nose out to do all of our gene expression analysis on all of our histology, and that he would much rather keep the whole snout intact and decalcify those snouts and then perform everything in intact noses. And here you can see a scanning electron micrograph of how that looks like, with this operculum covering the entrance of the epithelium of the nasal cavity, and down there is that rosette that I told you at the back, where the smelling happens. And this is just another example of what it looks like. So you guys can now imagine that when we did not put the tweezers inside and we didn't take this out, we were basically preserving all of this area that was surrounding the rosette, thanks to Pedro, who told us not to remove it. And when we actually started to look at just very simple H&Es, we were dismayed, because all of a sudden we realized that if we just leave all the wall that surrounds the olfactory rosette, this entire wall is covered in immune cells, as you can see here. These are all just lymphocytes. And we started to really go deeper and deeper into this question. So Ben and also Dong Feng, who was visiting from China, tackled this question for like 2 or 3 years, where we wanted to see if this, what we have now called organized nasopharynx-associated lymphoid tissue, did have or not any resemblance to those inductive sites that we find in mammals. And the first thing that we did, it was stain for different immune cells. So we stained for IgM B cells, IgT B cells, CD4 T cells, CD8 T cells, and then we also did a combination of those markers. And in fact, we were able to find all of these different subsets of B cells and T cells, forming really beautiful clusters. Here you can see all of these groups of IgM and CD4 T cells in the organized snout of a control rainbow trout. And this is what we need to quantify. So the organized nasopharynx associated lymphoid tissue is about 50%, 56% of CD4 T cells, 16% are CD8 T cells, and we have about 28% of B cells combining IgM and IgT. And we wanted to then quantify better what are the molecular markers. So this took a long time. We did laser capture microdissection, purifying or isolating RNA from only the O-nalt, and we compare that to the diffuse nalt, which is what we call here the tip, which is just basically an olfactory rosette. And we showed that the O-nalt was increasing expression for all T cell markers, TCR markers, as you can see, and IgM, but not IgT. And the most, exciting finding was that we were able to find expression of a gene called AID, and that expression was about 15 to 20 times higher in the O-nalt compared to the tip of the olfactory lamella. And this enzyme is the major enzyme that is responsible for the affinity maturation of B cells. Like broadly explained, it introduces mutations in the B cell receptor, and AID basically changes those receptors in a really random manner, but those B cells that are more now have a stronger affinity for the antigen will remain and progress through that cycle that I told you. So this is truly the marker that we were looking for, and we were very excited to see that that was going on much greater rates in the O-nalt versus the tip. So in 2018, I became an associate professor, and in 2022, I became a full professor. And I just want to add a lot of my journey here. By then, I had 2 kids, and it was incredibly challenging to do everything that we've done over the years and bring up a family. But I just want to say that all of the women in the audience, just don't let anybody tell you what you should do. Just do what makes you happy, and it is possible to have a great family and also a very successful lab. And I have now number 3 that just joined us 9 months ago. So I have a baby who is 9 months old, and yeah, they basically mean everything to me and brought so much joy to this scientific journey. So okay, I want to continue the last few slides to just update you on the very, very exciting times that we are witnessing in our field. And that's why I title my talk The New Era, because there have been tremendous discoveries the last 2-3 years. And these have really been an explosion of newly described lymphoid structures. The first of them is the ALT. And this was discovered by Julien Resseguier in Oslo in this paper by Dalum et al. And you guys can see that this amphibranchial lymphoid structure is also part of or is found in the gills and is right here in pink. At the base of all of these lamella, and is again, in this case, what they did here, it was labeled for T cells using a SAP70 antibody. So it seems that these structures are highly rich in T cells, and they're beautiful. All of these images, in this case, are in zebrafish, but they also work in other species, showing that the ALT was found there. Then only last year, the group by Anna Huttenlocher here in the US, working on zebrafish, had described this tessellated lymphoid network. And it's really interesting because it has only been done in zebrafish because, of course, they use a T-cell reporter for mature T-cells, the LCK reporter. But this was a really surprising finding, and I would love to know whether other species also have this massive network of T cells in their skin, and how do we target those when we are basically doing any immersion vaccination, for instance, because of course immersion vaccines should be stimulating these T cell responses, but also microbiota, of course. Then we have this another amazing paper that I was very lucky to be part of, also led by Julien in Oslo, and he coined this newly discovered organ, NELO. And the NELO is actually this incredible network of immune cells. Again, this is the same for sub-70s of T cells that links actually the gill lymphoid structures with the thymus and with this NELO, which is actually located in the pharyngeal cavity, so the lower pharyngeal cavity. And they did amazing 3D renderings. NELO has this kind of shape of the horns of a reindeer, if you want to, or a deer. As you can see, they run underneath the pharyngeal cavity. And again, this is a very exciting discovery, because truly what Julien's work have shown is that if we, and our work also on the nose, is that if we preserve the 3-dimensional structure of all of these mucosal organs, we are able to uncover much more than what we thought before. And of course, that has required much more advanced techniques as well as tools to be able to detect those immune cells in there. And then the very last one that just came out a month ago or a few weeks ago is called the ALO, which is the axillary lymphoid organ. This was only described in zebrafish in detail, but the paper has a few species in there, including a table where they show where is found and what is not. And you can see it is there, is this green structure here, right by the pectoral fin. It's full of lymphatic vessels and blood vessels. And in there you can see that, and this paper also shows a single cell transcriptome of this ALO, and yeah, very exciting because of course of the location of what it is, and it should be really exposed to pathogens that are trying to enter the body through the fins and through the skin. So I encourage you to check it out. There has been this massive explosion of description of new immune sites of mucosal tissues, but do we still know whether or not they're inductor and effector sites? We don't. And do we know if mucosal vaccines are able to stimulate these sites? We still don't. So because of the markers that I show you, the mucosal marker, gene expression markers that I showed you before, we did hypothesize that that organized lymphoid structure that we described in the nasal cavity remote trial could perhaps carry some resemblances with mammalian inductive sites that I show you, that they have germinal centers and where B cells become educated and selected. So we have a vaccination trial where we use our vaccine model, the live attenuated IHMB, and we did either one dose or 2 doses 4 weeks apart. And then we sample at week 6. All of these were done intranasally. And when the first thing that Ben did was to basically check how do immune cells change in the olfactory organ, particularly in the ONALT, following this primary or secondary immunization. And we were able to show that both primary and secondary immunization increase the numbers of CD8 T cells, IgM B cells, and IgT B cells, but mostly only following the primary vaccination. It's true that we only sampled 2 weeks after boosting, so this is something that we are kind of following up right now to see whether this is just basically the timing following the booster that accounted for these results. And at all points, we were comparing what happens in the non-organized sites. Sorry, I'm missing the stats here, but we were able to show some increase as well in IgM B cells in the tips, and some increases in CD8. Nothing was significant for IgT. So the critical question was whether B cells in those organized structures are doing something similar what we would expect to see in a germinal center reaction, which is B cells should be proliferating, as well as B cells should be being eliminated via apoptosis. So Ben did all of these EDU injections in vivo, and then Fen also, of course, both of them, and then quantify how many B cells were also EDU positive in our ONALT structures. And you guys can see that this was really exciting because we definitely saw increases also with the booster of how many IgM positive B cells were proliferating within the ONALT. And more excitingly, this was completely different to what we saw in the diffused NALT, which is the rosette in the tips. That was not happening over there. It was unique to the ONALT. The next thing that we wanted to show is whether or not molecular markers of germinal center-like structure were also found in these vaccinated groups. And we looked at CXCR4, we looked at TNF-alpha, we looked at AID, and again, very exciting results. We saw in the ONALT, but not the tip, that CXCR4 was increased, expression was upregulated. And while AID was slightly upregulated, about 3-fold, following one delivery of IHMB in the tip, so the diffuse NALT, the most exciting result was that we were able to see amazing increases up to 12 to 15-fold in AID expression in the ONALT following one round of vaccination. And it was kind of interesting that after the booster, we didn't see that, but again, this could be due to our sampling time points. And we didn't see anything for TNF in terms of gene expression changes. So the next thing I told you is that it was very important to know whether apoptosis was happening in the ONALT. So the first thing that we did is to check at a molecule called BCL2, and this is a brake on apoptosis. So we hypothesized that this brake, if apoptosis was going to occur, this gene should be downregulated in response to vaccination in the ONALT, but not in the tip. And that's exactly what we saw. We saw a downregulation of BCL2 following one round of vaccination in the ONALT, but not in the tips of the lamella. And actually, we then did microscopy to confirm this, to quantify how many B cells were apoptotic in the ONALT versus the tip. And we were very excited to show that indeed there was an increase in the number of IgM positive, caspase-3 positive cells in the ONALT. And that was not true in the tip, as you can see on this right-hand side panel. So one of the things that we are still working on, and have a whole new grant on this, thanks to USDA and NIFA, is that we want to optimize interval times. So right now, what we know is that there was a trend towards increased neutralizing antibodies in the boosted group when we sampled 2 weeks apart. And remember, these were boosted only 4 weeks apart. But right now, Ali in my lab is basically having thousands and thousands and thousands of fish that are being vaccinated, not just intranasally, but by immersion, injection, etc., in Idaho. And what we are doing is trying to see whether 4 weeks is maybe not enough, but whether if we prolong interval times, maybe we can favor definitive maturation of the immune response, and hopefully can tell you more about this the next time we see each other. So one of the last questions that I want to bring up on the questions in the field is how do we leverage this knowledge when we are trying to vaccinate really young animals? And we proposed in our grant, and we've now determined this, and Ben has now finished this piece of the puzzle, is that ONALT actually is not formed during ontogeny until rainbow trout are 1400 degree days. However, adding to this puzzle is that we know that we can intranasally vaccinate fish much, much earlier than 14 degree days. We know 1400 degree days. We know that if we vaccinate trout around 350, 370 degree days, so they are very, very small, we still can induce incredible high levels of protection through nasal vaccination. So the question is, does it matter, right? Does it matter that we vaccinate before or after these OMALT are found? And again, we just basically running these experiments as we speak, where we just vaccinated fish before 1400 degree days that they don't have the ONALT. And now we are about to wait for them to be 1400 degree days and perform the same whole experiments. And then we will challenge afterwards to see whether this makes any difference in terms of protection. We are going to do very long-lived, I guess, studies. So we will challenge maybe 4 or 5 months from now. And we will also track antibodies and the affinity of the antibodies. So in conclusion, I just want to wrap up to say that mucosal immunity is very, very complex, and we have so many unknowns, and that's why it has been such an exciting career so far, and I can't wait for what is to come. I also want to say that it is really important that we continue to invest in basic science, because we are able to really inform the things that matter for industry. And I really, really care about that. I worked in a fish farm when I was a student, and I love to be hands-on and seeing what the issues are for farmers. So for me, it's really rewarding to be able to translate our findings. And that we were wrong for many, many years. We always thought that there are no organized lymphoid structures found on mucosal sites, but at least structurally, we know that they are found. So it's okay to be wrong for many years. We just need to be challenging ourselves and re-questioning ourselves and our hypotheses. And I just want to say, of course, the best part of science is the people we meet on the way. And I want to also just do a shout out about a little bit about inclusive excellence, and saying that it's amazing how good science can come from anywhere, from any minds and any countries and institutions in the world. And we should definitely foster bringing those diverse perspectives to the table, and support each other, particularly women supporting other women. I think it's very important. And with that, I'll take questions. Oh, so thank you to all my collaborators through the years, and also USDA and NIFA for unwavering financial support, many, many years of support. I really appreciate it. Thank you.
That was a spectacular presentation. There's a virtual round of applause going on right now for you. Outside of my wheelhouse, but a fascinating topic. Folks, if you have questions, feel free to unmute yourself, or you can drop the questions into the chat and we can ask them that way. All right, we got one from Taylor here. She's wondering if there's any evidence for IgD playing a role in microbiota homeostasis in IgT-less catfish.
Hi, Taylor. So good to hear from you, even if it's just on the chat. So the only paper on IED and microbiota is in Rainbow Trout from Carolina Tafaya's group, the Cell Reports paper, which is incredibly good. I know she's following that up. They have been able to purify IED from gut mucus. So stay tuned for that. But I would say definitely the catfish people, you catfish people should tackle these questions. And this needs to be done because it's a very, very, I think, important question. Even knowing what is the concentration of IED in the mucus of catfish and IgM? Like, we don't even know the most basic biochemistry, glycosylation, polymerization. We don't know any of that. And I think that's waiting to be done. Other questions? Yeah, she's a new catfish team member. I know.
Yeah, Irene, it sounds like you need to come to Stoneville and visit.
Yeah, I would love to. I haven't been there yet, so I will be delighted. Sounds like a great facility as well, so it would be really cool. Yeah, no, IUD it's hard, and the thing about IUD is that it's so little known in humans that whatever you discover in fish, I think it really advances just like knowledge in general. So the very first big paper on IED was in Nature Immunology. And if you guys remember that, actually that work was done in catfish first. And then the catfish folks who discovered that IED can bind directly basophils, then they talked to human and mouse people, and then they put together this big paper in Nature Immunology. But truly, that was done by the Mississippi team. In catfish IgD, but they didn't look at microbiota at the time. They were just looking at binding to Fc receptors in other immune cells. So Greg has a question. Hi, Greg. Are there different roles for allo versus nello in the immune response? So this is all waiting to be done. In the allo paper, they didn't do anything. I mean, they did a little bit of functional stuff with like quantum dots to show that they move around. I think they probably have more stuff. They did a little bit with a lymphoma actually model. They show just B cell changes or in a lymphoma model. But in terms of applied to aquaculture, it hasn't been really done. So I would love to know what happens there. The Nello paper, Julien did have stuff on infected fish. And I'm sure he just got a big grant to continue on this. I'm sure he's working on this. As we speak, we are looking at just basically whether Nello and ONALT are core. So the development is something that I'm really interested in, whether are all of these lymphoid structures appearing at the same time during development, or are there different sequences of ontogeny there? Because I think that is really important for mucosal vaccination, right? Like, is it the Nello is the one that appears first, then maybe stimulating the pharyngeal cavities where we should be aiming for first. So there is all of the functional stuff and more applied stuff is there to be still, waiting to be done. But I should maybe ask Julian about the Nello vaccination part.
Any other questions out there?
Hope you're okay, Greg. I haven't seen you for a while. Thank you for coming. Any more questions? And if anybody has questions about protocols or like, how do you do this or how do you do that? Like, I'm super open to, like, you guys can email me or call me and I will share protocols with you. Many of our protocols are on our website, but I really want to, again, emphasize like open science and collaboration and sharing knowledge is really important. So.
That's great. And that's a great way to end it. Thanks again for sharing your story and experience with us. It's fascinating work.
Thank you very much for having me.
Yes. All right.